Hi, Praveen. Welcome to Idea Spring Founder in Focus podcast. Lovely to have you here. The reason we are doing this is to talk to our founders and other members in the ecosystem to understand their journey and the reason they started the company and then what would they like to achieve going forward, right? So I think these aspects will be good for our listeners and to learn from, and also for other people to understand what you're going to do with your company and what's your ambition and other aspects of it. So just to kickstart, quick introduction about yourself and how did you get to starting KCAT?
Thank you, Nagarand, for the invitation, and I thank you very much for this opportunity. This is Praveen, and I am a computational biologist. My specialization is in protein modeling, quantum chemistry, and machine learning for developing or engineering proteins for biocatalysis. I have two other founders in the company, Mr. Gladstone Sigamani, who is an artificial intelligence specialist, and Dr. Rupa, who is a biochemist. So we joined hands together in 2019 and started this initiative of KCAT Enzymatic, which focuses on developing enzymes for biocatalysis. We started this company with two people in a co-working space, and now we are 56 scientists spread across different fields like molecular biology, artificial intelligence specialty, and quantum chemistry, fermentation specialist, providing end-to-end solution for biocatalysis.
Yeah. So you said enzymes for biocatalysts, right? So what does it mean actually in a scientific or technical terminology?
So generally, pharmaceuticals, nutraceuticals, and agrochemicals make their chemical entities or compounds using chemical processes. And these chemical processes are highly entrophic. So they consume a lot of energy in making them. So they work at high temperature, pressure. And so it's not good for the environment. It's not good for the people who are working there. And also they are not good in making they're not good in giving good yield of the product. So they give on an average of 97, 98% yield of products that they are forming. And very importantly, they are not good in making chiral chemistry, which is if you're making a mirror image of a molecule, where in many cases we say that an R-specific outcome or R-specific molecule enantiomer is very crucial for the activity of a particular disease to target a specific disease, then you make only R-specific outcome. Now, what happens in a chemical process is they also make S-specific outcome, which becomes a side product, which may not be good for the biological activity. It may become toxic for the body. Therefore, it's not a good idea to make chemical products. For some of the APIs, it's not a good idea to use chemical processes to make APIs, active pharmaceutical ingredients. And therefore, people started using enzymes. Enzymes are milder. Most of the reactions that are done in our body are enzymatic. Or in fact, say all the reactions that are done in our body are enzymatic. And they, in a bacterial system, if you pick any sugar breakdown, it is done by enzymatic approach. So these enzymes are proteins and they act as catalysts to accelerate a reaction. And since they are milder, they happen in a physiological condition. You can take these enzymes in a room temperature or in a room and you can conduct the reaction. So as I told you, it works at normal temperature, normal pH, no pressure involved, and therefore you conserve a lot of energy. The very moment you start conserving energy, it becomes cost effective and eco-friendly. And you ask the next question where, why were enzymes not used?
Correct. What stopped anybody from using enzymes
before, right?
Before, yeah.
So they started making chemical entities in a chemistry lab. So enzymes came into existence only in 1970s, where they thought enzymes can be used for making these kind of chemical entities. So scientists invented methods to use these enzymes in a laboratory scale. But the very moment they started scaling up to an industrial scale, it became very difficult to scale up.
Control the experiment
or scale up the process itself. There were no yield they were much less at yield and therefore not cost effective. More enzyme concentration was used for making the chemistries, and therefore they were not cost effective in making them. So there were a lot of critics from the chemical world, which makes chemistries using chemical processes. But luckily, there were a couple of scientists, Professor Arnold Francis, who received the Nobel Prize in 2019, who discovered the process called as directed evolution for making enzymes. She proposed that enzymes can be genetically modified to accelerate a chemical process, even at a bioreactor level. So after engineering, you incorporate these changes in the genome, in the genetics of the enzyme, which will translate to the protein. And this protein now becomes robust enough to work in a bioreactor to give the product that you want at a higher substrate concentration, at a solvent which is not used to by the enzymes, and which is working at different pH, and also at varying temperature.
Understood.
So all these are possible with very high yield. Another aspect is it is very, very good in making chiral outcomes.
What do you mean by chiral outcome?
What I mean by chiral outcome is, as I told you, the substrates or the products are made of mirror images. You have S form or R form, which is like a left hand and right hand. They may look similar, but they are two different structures. So in order to make only specific form, enzymes are champions in that. You can use enzymes to make specifically R form in many cases, or specifically S form if that is required. So you can engineer the enzymes to make 99.9%. So there is no side product form. There are no toxic outcomes. There are no, unwanted stereoisomers that exist in your end of the reaction, which could be poisonous or which could be toxic for a biological function.
So if the enzyme manufacturing process is so environmentally friendly and so cost effective and so pure, why aren't we seeing a lot of companies doing that? And more specifically, why is KCAT uniquely positioned to solve this problem and manufacture enzymes in bulk?
That's an excellent question. See, many think that developing enzymes are as complex as a drug discovery process. That's because you have so many permutation combinations that can be incorporated, which scientists don't know how to actually incorporate them to get the best enzyme that works in a bioreactor. So far, scientists have delineated the mechanism of enzymes only pertaining to the active site of the enzyme. Active site is only a small component of the enzyme where the substrate enters inside the active site and gets converted to a product. And there's a process called as quantum chemical calculation, QMMM simulation, which received its Nobel Prize in 2013, using which people have delineated kind of how an enzyme substrate is converted to a product in an enzyme active site. So this is a place where the enzyme comes in physical contact with the substrate. But there is a whole lot of other regions of the protein and domains of the protein which governs the reaction of the enzyme, which people don't know about. And that is where mutations are incorporated to stabilize and to make a robust enzyme that works in a bioreactor. So far scientists have not identified or delineated a step-by-step process or a streamlined set of protocols which can be used for engineering enzymes. So it's all based on serendipity, permutation combinations. It's a brute force based method that you engineer enzymes. So it becomes very complex, cost effective, cost intensive, resource intensive, and takes sometimes years together, and you end up sometimes not making an enzyme for a specific project. That's why many of these companies shy away from these kind of projects. So at KCAT, we have developed technologies and methods, which is completely AI-driven method to develop enzymes.
So help me understand that more. What do you mean by AI-driven method to develop enzymes? Because I've read about protein folding and then AI now trying to solve very complex problem of protein folding. So how is that something very similar, or this is something very different?
It's absolutely very different. I totally accept that AlphaFold has cracked the problem of protein structure prediction. But so far, we don't have methods to understand the functionality of proteins. So I would like to start from my history, where I started my career in target deconvolution studies. And one of the protein that captured my attention was so-called actins. which actually forms the cytosol, a skeleton of your cell. If you take a cross section of the cell, the majority of the cell is made up of actins, and these are also enzymes. And I wanted to understand how this also acts as enzyme, and implemented that quantum chemical simulation and tried to understand the whole theory of mechanism of enzyme action. Now, while doing so, what I also understood is it's very difficult to develop an enzyme for industrial process. So there came a couple of projects which I worked. One of the specific projects that I worked was on penicillin GSLase, which is involved in making beta-lactam antibiotics. And I was quite successful in developing enzymes by incorporating mutations using, very primitive algorithms, and was able to improvise the activity in the industrial scale. But that was not enough. If you want to start something from scratch and where you don't know the activity of the enzyme, and the enzyme in your cellular system works only on certain natural molecules, which is existing in your body or in a plant. But if you take them and put it in a bioreactor and ask them to work in a semi-synthetic or a synthetic substrate, they don't work at all. So in order to derive those functions, you have to develop sophisticated algorithms. And when these enzymes, which unlike any other AI methods that you use for developing the structures or solving the structures, these enzymes are very dynamic. They go through very different conformational changes to convert a substrate to product. So in order to study those dynamics, you need sophisticated methods and processes. Of course, there are methods and processes which scientists have evolved, but as I told you, we don't know the functionality of these amino acids or domains which are far away from the active site. So we developed this so-called 7D grid technology, which captures information at 0.1 Armstrong resolution across the enzymatic reaction and across the enzyme substrate system. So with this, we are able to capture the whole energy landscape that would exist, that would happen, that would form during the whole reaction, and able to identify spots which, when engineered, can give improved activity. And what we have done is, since this is a lot of information, and if I'm able to show this as a grid, where it's a 3-dimensional grid, and the 7D grid is a 3-dimensional grid, and this grid is divided into grid points spaced at 1 Armstrong resolution, and each grid point is defined by so-called fragment molecular orbitals. So orbital information is captured across the enzymatic reaction. And after capturing, this is integrated into an AI-based process. Gladstone has established diffusion-based, denoising diffusion-based probabilistic model. And he has also established message-passing neural network-based model, using which we are able to capture information and identify specific regions where it can be incorporated. And we are quite successful. We initially...
you have patents or something around this now?
Of course. We have close to 3 patents. And initially we got something...
3 patents or close to 3 patents, or you have 3 patents?
We have 3 patents. We have 3 patents on it. And 3 patents only on the technology. We have product patents. We have filed close to 24 patents otherwise. But the technology patents is around 3 patents revolving around this particular concept that I am right now discussing. So once this is implemented in the AI, it is able to give you a very good agreement with the experimental data. So back then, we were trying to establish so-called structure-activity relationship models with enzymes. So we used a regular regression, and then we implemented neural network-based regressions, and then we implemented MPNN and different other NN models. Now it is giving close to 80% accuracy with what we predict. So this is going to reduce from what in lab, we had to establish close to 40,000 of lab experiments to derive a working enzyme. We cut down to 200 to 300 experiments. So we can cut short this time by, what would have taken 18 months to just 2 to 3 months. We should be able to finish the whole project.
Lovely. Starting
from scratch.
So that way you can identify which enzymes are best suited to solve what problem, and, in the lab first, in the simulation first, and then bring it to the lab, and then hence get the product to the market faster.
Absolutely.
Yeah.
Absolutely.
And that's one of the key differentiators you're looking at.
Absolutely. That's the key differentiator. And moreover, in the last 4 years, we have developed close to 17,000 enzyme variant libraries. that helps us a lot because that becomes the training data for us to develop new enzymes. And we have also established a lot of data from the open source, from the literatures, publications, and also from the patents with which we are able to establish enzyme activity within a very short period of time.
So in your journey in KCAT, where are you now, with respect to challenges that you still think you need to solve, both from a science perspective and from an engineering perspective, right? And I'm saying engineering, I'm talking about enzyme engineering, manufacturing and so on and so forth, that I classify as engineering. So what are some of the key challenges you think is still ahead of you in science and key challenges still ahead of you in engineering?
So one specific problem that we are working with the European Union is a fluorination problem. Close to 25% above of the APIs are made up of fluorines. And the chemists, they define this as dirty chemistry because it's very difficult to make organofluorine molecules. So if you're able to make it using an enzymatic approach, it'll create magic in this particular field. And most of these fluorine molecules are very active molecules. Many anti-cancer drugs, many anti-diabetic drugs are fluorine plugged molecules, which is very active. So our objective is to, and it's very tough enzyme, it's a very finicky enzyme actually to engineer. So we have tried many rounds of experiments and we have got some good success. And so far we are able to plug fluorines into the phenyl group of some of the molecules that we have established in our lab. So we envision to make this as one of the biggest products of KCAT. So it's challenging. It might take a while, but we will be there very soon, we believe. We're also working on other challenges such as engineering enzymes for developing mRNA vaccines. So future is going to be mRNA vaccine, and we need very precise enzymes to make the specific mRNA that you're looking for. And these days, the present days enzyme are not so efficient. You have, again, cross-reactivity, you have noise in the reaction, you have different other mRNAs which is not needed, and it might be immunogenic, double-stranded mRNAs and so on. So we are developing enzymes towards it. Other areas that we work on is plastic degradation. We care about environment. We don't take it as a mainstream development, but we also are working on enzymes which can degrade plastics.
Understood. So now, when you started KCAT, obviously you got a lot of grants, and then, IdeaSpring also is participating in a journey, and we gave you the first institutional check. What are some of the key milestones you're looking to achieve? Where do you want to be the next 2 years? Where do you want to be the next 5 years from a KCAT company perspective?
So, first of all, I think we were in the right time in the market, I would say, because a decade before, I don't think the Indian pharmaceutical company would have recognized this as a very important field. Now, FDA poses stringent rules to make most of the chemical processes to enzymatic processes. And it becomes easy for even small scale and large scale companies to get FDA approval if they have enzymatic processes in their reaction. So while we started this a couple of years before, we got the first VIRAG grant for developing a transaminase enzyme for an anti-diabetic drug. And we successfully completed the project. And we have an orthogonal enzyme. What I mean by orthogonal is orthogonal from nature. So it's a fold that doesn't exist in nature. And we made completely an ab initio fold that was developed in the computer. And we are able to now convert it, becomes a robust enzyme which is able to convert a prochiral ketone to the product, which is a anti-diabetic drug. Now, we were working on the last 1 year for scaling it up, and we are quite successful. And this enzyme is almost giving 200 grams per liter of substrate conversion, which is commercially very crucial and important for Indian customers. So this is kind of breaking the monopoly, I would say, because this was mostly imported from outside of India. And now we are having an enzyme which is completely indigenous, made by India, and it will be used in India. And we also plan to export this outside to India. Now, with IdeaSpring's fund, it was very instrumental for us because we were able to establish our own laboratory. And now we have well-established molecular biology, microbiology, flow biocatalysis, and analytical chemistry laboratory, which now gives us end-to-end, which makes us an end-to-end protein engineering lab. So we are able to showcase at least 1 kilogram of substrate to product conversion that fits the cost window of the customer. And that interests the customer. And immediately they look for scaling up. They're ready to invest on manufacturing of the enzymes. What we right now do is contract manufacturing. They're ready to invest on this contract manufacturing of the enzymes.
And you mentioned briefly Indian pharmaceutical industry, and then you also spoke about export, right? So my assumption is you're building a company out of India that can, in fact, manufacture enzymes that can be sold across the world, right? So that's where we are going. But initially, you're focusing on the Indian market. Is that the right way to put things? What's your ambition here?
Yes, but we have done projects for companies outside India. So we've done projects for innovator companies. I think I can mention them. We have done projects for Pfizer, and we have done projects for different other top Indian companies that we work with. Now, these companies are now in talking terms with some of the important molecules, which is in the third phase of clinical trials. Now, that is where I think KCAT will have a breakthrough. So we need to work on projects which is going to be the turning point of the company, turning point of making a molecule that will work on an innovator's molecule. So that's one important business.
When you say innovator's molecule, what does it mean?
So the Indian companies are good in making generic molecules. We don't have an indigenous drug on our own. Most of the drugs are made by the Western world. And anti-diabetic, anti-cancer drugs are made by the Western world. But they out-license these manufacturing or chemical synthesis of these molecules to the Indian and other part of the world. And along with it comes the other components, which are the catalysts, which are also, most of them are patented and developed by the Western world or the Chinese market. So in order to break them, you have to come out with orthogonal processes and enzyme technologies, or you have to start doing a project with them where it's in the clinical stages. So that's where we are talking to them. Otherwise, it was developing projects which are already existing in the market, APIs, which we've already done some projects with the innovators. But right now, we are talking to them with molecules which are in the third phase of the clinical trials.
So you also received NIH grant, and I know you're collaborating with Tufts University. Can you just elaborate on what is the collaboration there and what problem are you trying to solve there?
We are working on a problem which is on a therapeutic enzyme. We were working on an enzyme called as PAL, phenylammonia lyase, which is involved in treating inborn errors. So it's an interesting molecule, and we are developing processes and methods, specifically also engineering the enzymes so that with less immunogenicity and is able to metabolize the amino acids, specific amino acids that we are working on. And in fact, we are the only company to receive this award as a co-awardee. And I think in the future, we envision to develop multiple therapeutic enzymes. So that is one stream of area that we are looking at, developing therapeutic enzymes. And the other stream that we are looking, as I told you, is plastic degradation. And we are also working on pharmaceutical waste management. And these are a couple of areas that on a broad level we want to look, focusing specifically on a battery of intermediates and APIs that we have received in the last 4 years from different customers, which are low volume, high value molecules for which we are developing enzymes. And these are across different areas, not only pharmaceuticals, it is also nutraceuticals, agrochemicals, and we are also working with L'Oreal on a specific molecule. And it's a very famous molecule of L'Oreal. And if when we have a breakthrough, I think it will be one of the turning points, like what I told you with respect to pharmaceuticals, when we make a molecule which is in third stage of clinical trials.
I think that's where you'll have a hockey stick.
Yes.
In terms of innovation and growth.
Yes.
And what other important or exciting enzymes you're working on where you're saying if this works, it's going to have a significant impact?
We have worked
we already have 6 enzymes, which are FDA approved processes where our customers are using these enzymes for producing their end product. And one of the very crucial enzyme is an enzyme which is used in making antidepressant. So this particular enzyme works on an amino acid which is completely insoluble, an insoluble amino acid. So therefore, it is very difficult to make it through a chemical process. If it has to be established through a chemical process, you need 1,000-liter reactor, 1,000-liter reactor for making 1 kilo of your end product. Now, through the enzymatic process, we have developed a process which can be done in 5 liters. So 200-fold of reduction in volume. So you can think of, setting up the reactor infrastructure and also the manpower. It can now take place in a room. What you thought, you need a factory, it can take place in a small laboratory where kilotons of products can be made using this process, this enzymatic process. So the good news, the good thing about that engineering that KCAT has learned from this project is that we were able to develop an enzyme that works at very high pH, extreme pH. So this enzyme is capable of working at pH 1, which is acidic, and pH 11, which is basic. So only at that pH, this amino acid is soluble. It brings down the volume to 200 folds. It's soluble in just 5 liters. What is then soluble, at pH 7 or 8, which should be at 800 liters. And chemical processes are only established at pH 7 or 8. It can go up to 10, which again 500 volume of the reactor. And we are also working on immobilization of these enzymes. And the good thing about these enzymes are, a tinge of enzyme is enough to make the whole reaction work. So 0.1% of enzyme is enough to convert the whole process, which means 1 gram of enzyme can convert 1 kilo of your substrate to product. So you don't have to spend much on them. Yeah, unlike the chemical processes, you need very little amount of enzyme. Only challenging factor is developing that enzyme to work at that titer. So you keep engineering to make it work at less than 1% or 0.1%, which is just 1 gram to make 1 kilo of your product.
Okay. So I think my final question is on your vision for KCAT. I know you, Vlad, and Rupa are working fervently in bringing new enzymes, new processes. But what is your vision for KCAT? When will you feel happy that you have delivered something?
So, I had this urge of starting something like this in India way back in 2013. When I first attended a conference in 2013 in Japan, it's an enzyme engineering conference, a renowned conference. And most of the players, or in fact say the only paper that was selected from India was my paper. And 99% of them were either from Western or from the European market and some from Chinese. So I had this vision to start this year, and we started this, and now we are going good. Now we want to break this, as I told you, the monopoly, and we are already doing it, and to get new projects out there in different dimensions, in different facets of enzyme engineering or protein engineering, and to make India the hub for specialty enzymes. That's the vision of the company.
Okay.
So if we are able to get the next 10 enzymes in the market across the globe, and we already see many of the European players are contacting us for the enzymes that we have made, specifically transaminases, ketoreductases, nitrilases, and hydroxylases, and some of these cytochromes, which are very interesting enzymes. So far established in the lab, which can do something called as carbene transfer, which can do carbon-carbon bonding, which can do silicon-carbon bonding, which are just available in the laboratory. We want to scale it up and see if this can work in the large scale.
Lovely.
Yes. So This is the ambition of the company. Of course, the revenue and growth is there, but I think when we make these scientific breakthroughs, which from the lab scale to the industrial scale possible, then I think we'll be top of the world.
Yeah, as you know, IdeaSpring goal is to
come in very early in very innovative companies, and we come in pre-revenue, and we're really excited when the founders have these kinds of breakthroughs and build something from India for the world, and we really look forward to that. And with that, thanks a lot, Praveen, and we wish you all success in what you're trying to build. And we look forward to being with you in this journey and build, like you say, a company in India where we have the top enzymes that can be bought from here. Thanks a lot.
Thank you, Nagaran. Thank you for IdeaSpring for motivating. Thank you for this opportunity.
Thank you.